Yeah, I mean, I really thank everybody for coming. It's a wonderful. I see familiar faces and familiar names, which I know are interested in DNA, resonance, and consciousness. Okay, now I invite a new next speaker, which is me, and I will start my presentation. I'm Max Rempel, and I will speak about biophysics of consciousness and DNA resonance. Can you see my screen?
Yes.
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 So I should highlight just a sec, I need to move it away. I should highlight the website dnaresonance.org, and you might know it from because you came here through it possibly. So this talk is about Freire model, and I published the whole. It's first is there's a chapter which is self-published. It's not published yet. It was invited to a book, but it's not published yet. So if you go to DNA Resonance website, you click on, so video presentations will be here, full text publications, click on the full text and you get to that. And this Freire model is on the next page. It's called Rempel Consciousness Freire 4M. It's a big chapter, which I will try to squeeze here. The whole problem of connecting consciousness and DNA is huge and complicated. It's not trivial. We don't know a lot, and a lot of things are transdimensional. So we really have very few ways to reach there. But there are paths of connecting back and forth, and I will show a few connections. I will start from the model that we published a while ago, which connects DNA in the nucleus. So DNA is in chromosomes in the nucleus of the cell, and Khamerov and Pentros suggested that microtubules are important, and I think the argument is very strong that microtubules are transmitting and participating actively in the work of cells, and especially in the work of neurons transmitting information. So that's Khamerov, and he is basing that idea on the action of anesthetic drugs and on geometry of microtubules and on the function of microtubules. So what we suggested is that DNA vibrationally is coupled to microtubules. Microtubules are in cytoplasm, and they come close to the nucleus, and actually they connect to the nucleoli, and they plug into nucleus vibrationally this way. That's a hypothesis, not proven, but if you look at the photographs of microtubules, then perfect candidates for actually transmitting information. And the next idea is that DNA in one cell, that's the problem, how DNA can talk between the cells vibrationally. So DNA in the nucleus of one cell is connected to the DNA in the nucleus of another cell through microtubules. They vibrationally connect. They go to connection between neurons, which this is axon, and the connection is called synapse. So in synapse, they don't touch each other, but they come, if you look at the microphotographs, they come very close to each other. So there is a gap. And this gap is called synaptic junction or synaptic gap. It's a major, well-studied gap, synaptic gap, which is regulated by pretty much all psychically active substances. So this is well studied, and all the famous dopamine and serotonin and GABA and other transmission and receptors are regulated through drugs in this gap. So I learned this idea from Richard Alan Miller that resonance property of this gap probably defines a lot of qualities of the mind and psychology and emotions. So it's not only chemistry, possibly it's a resonance of the gap is the key to consciousness. So that's the simple model. that the DNA of the body is connected into one unified vibrational system. I will just go through highlights. The next topic is what could vibrate in DNA. And here we pay attention to the genome, So genomic sequence is known. It's 3 billion base pairs. And if you each chromosome is about 10 centimeters. If you stretch all the chromosomes in one line, it's about 2 meters. It's all packed in a tiny nucleus, which is about size of 5-7 micrometers. And if you look at the genomic pieces or genomic sequences, the well-studied part of the genome is called protein coding exons, protein coding part of the genes. They're called exons. And its total protein coding part of the genome is 4.5. So that's what is studied, well studied. The rest is called junk DNA or non-coding part of the genome. And we believe that this is the part which is actually takes part in resonance and consciousness, the non-coding 95.5%. And from this part, introns are also part of the genes, and they're not translated into proteins, but they're part of the genes, the insertions in the genes. And repetitive part of that is 24%. Why are we talking about repetitive? Because for resonance, you need to have multiple units to vibrate and resonate together. So repetitive part is what we think is actually responsible for resonance and structure and structural properties of chromosomes. And the rest of the genome, which is 46%, is intergenic sequences. And of them, repetitive is 28% of that. So total together, it's over about 55% of the genome is repetitive. So that's huge. And that's, I think, the key to consciousness and to the function of resonance function of the DNA. Jump into the next topic is the proof that DNA is actually genetically important. We know that children are similar to parents. Even if they were separated at birth, they still inherit the properties of parents. So we believe DNA is important. The mainstream science believe that DNA is super important, but Sheldrake, Rupert Sheldrake, questioned the importance of DNA, and he demonstrated that actual estimates of contribution of DNA to the phenotype, the heritability, is hugely inflated, and I agree with him. So the most of the studies that prove that DNA is important, they use twin studies. So they do twin heritability estimates, and this, in my estimate, in my table, describe the details are described in the chapter, but basically they are pretty much double or even more, maybe they triple the actual genetic heritability. So true genetic heritability is less than a half. The rest of variance is something else. We can talk about that, but we are not only genes. There is other factors which contribute to the phenotype variance. Okay, so inflation in twins, I believe, is happening because twins synchronize in their womb. So they develop together and they become synchronous in the womb. And that's why they are more similar to each other than the fraternal, the identical twins are more similar than fraternal twins. So genetically identical twins are more similar because their DNA synchronizes. So that's why the twin study overestimate genetic heritability. So it's called missing heritability gap. It's well known, and it's kind of swept under the carpet by geneticists because it helps them to get more funding. If genetics defines, say, 80% of, say, in schizophrenia, twin studies show 80%, but If you actually measure in GWAS studies, it's much less. It's less than 40%. Usually it's 10 to 30% heritability. So, but DNA is still important, just not that huge. It's less than half, maybe a third of the variance. But still, in that how do you call it? In this in this true heritability, it is resonance, I think, contributes a lot. So the non-coding part of DNA contributes a lot, and proof for that is that if you look at the non-coding part of the genome and conservation in the evolution, it is nearly it is also well conserved. There is between the genes there are conserved sequences that show that DNA is important. That non-coding DNA is important. Okay. Next, I jump to the next idea is nature of consciousness. And here I just accept the model which is known for forever. It is in all cultures that there is a universal consciousness, and that we that personal consciousness is part of the universal consciousness, and that the mind so that's the human body, that the human body is part of the universal consciousness, and that it cannot be separated, that the illusion of separation is just illusionary. And in this picture, I'm trying to place the DNA in it, and in the body, I place DNA outside to show that this, in my hypothesis is that DNA is the actual interface between the mind and the universal consciousness, and much of the mind exists outside of the body and is connected to our physical conscious mind through DNA. So basically, through resonance and other information transfer, the universal, the external subconscious mind is connected to the internal mind. So that is part of the Frere model. And the second part basically says that the outside world, this green outside world, is created by as a simulation, is created from inside out. It's not only my model. I just accept the well known idea popularized in the movie Matrix that this is a simulation. I don't say it's computer simulation, but it is natural simulation in which the outside circumstances are created from inside out. Proving it is pretty much impossible, because that simulation, it seems to have a rule that it protects the illusion of separateness and protects itself from being discovered. So from inside out, you cannot really prove that you live in simulation. You can have a personal experience, but sharing with others is prohibited. And there is a lot of protective programs that prohibit us from proving to others that we live in simulation. So the key from this slide is that physical body has a skin, and the skin separates us from outside. And it is an illusion that mind is located in the body and that mind is located in the brain. And this illusion is created by through resonances in DNA. Okay. then there is a dilemma which is pretty much also unsolvable. I just kind of plotted on the picture that we know that consciousness creates, no, the claim of this theory is that consciousness creates the body and creates the brain. So consciousness creates the brain, and brain creates consciousness. And that dilemma is solved by dividing to universal consciousness and individual consciousness. So the brain develops through experiences individual consciousness. But that is all part of the universal consciousness shown on the previous picture. The next idea is we need to solve this riddle to explain the true role of DNA in consciousness. So it's unavoidable. The next idea is to understand the flow of time, the illusion of flow of time, and the free will. And here I demonstrate, it's called bump and go car, and you all know them. They were from the 50s and 60s. These are the cars which go, and when they bump into an obstacle, they rotate the wheels inside, and then they choose a new direction as if they were self-driving cars. So that existed even before electricity. So that's one of the examples of self-driving behavior. So to make choices, you can use this mechanical or largely mechanical model, which makes similarly choices. So what is free will choice? It's the choice which is made in the absence of full information and under the pull of time, under the pressure of time. If you had unlimited time, you probably would choose the perfect solution. If you had unlimited time and unlimited information, you probably could calculate the perfect solution for any problem. But because of the pool of time and limited information, you have to make so-called free choice. So there's a free choice and pool of time are critical. And again, I published a paper where we suggest that DNA plays huge role in creating the illusion of pool of time, illusion of time. The next idea is idea of fractal rendering. So rendering, you probably know from computer games, where in a computer, I don't know if you play computer games, but at least you watch people playing computer games. And in many games, they are multiplayer games, so multiple individual consciousness are in the game. And you walk through the world or fly through the world, drive, and as you walk through the world is created in front of you. So that is idea of rendering, and it's also pretty well known. The idea is that the reality doesn't exist until you pay attention to it. So as you walk through the world is rendered as if it was stable. But that idea of rendering is huge. And the next idea of fractality is that you are a fractal part of the universal consciousness. You're a fractal fragment of universal consciousness. So the idea of fractality that I'm coming back here, that the main trick to understanding the role of biology and DNA is to understand that the illusion of body, illusion of mind, and illusion of time is created by separating us from the rest of the world. And what's inside the skin is a fractal mirror of the outside world. So what basically it's a mirroring, it's transformation, mirror transformation, fractal mirror transformation. What's inside out, what's inside is a mirror of what's outside. So that idea is, I guess, is it has many names, but one of them is monism, another one is solipsism, and it is known for a long time. Here I just wanted to combine to bring four main players. DNA sequence contributes to mind through many ways. And the main claim of this presentation that DNA dance is how that happens. So DNA dances and produces thinking. And then also DNA contributes to morphogenesis, the shape of the body. And also it produces, through the shape of the body, it produces the brain and the mind and the personal individual consciousness. Okay, next I need to suggest that it's impossible to understand the role of DNA and in consciousness without looking at glitches in the matrix or glitches in a materialistic model of view. And one of the key breakthroughs, I don't know if you're familiar with that, happened just recently. A year ago, actually, it's like 9 months ago. Telepathy tapes came out, and they are led by Kai Dickens. And now they are created a big movement pretty much all around the world, because it was discovered that telepathy is real, and it's measurable, and there are very good telepaths around. So the main discovery was that autistic, non-speaking, non-verbal children and adults are usually telepathic, and when the normal people, they're called I'm blanking on the name, neurodivergent versus neural something. When normal people start teach non-speaking autistic children typing, then this telepathy is pretty easy to measure and prove. So telepathy tapes is a documentation of that community growing, discovering telepathy in non-speaking autistic population. And this population finally got a voice. They publish books, they make videos, and in many cities now there are communities where people finally discover the telepathy. And if you know Strugatsky's prediction in book Гадкие лебеди, I don't know the translation, bad swans, ugly swans. The idea that the humanity evolves into the new species. So the idea is that this new autistic population is the next branch of human evolution. They're genetically different and they possess telepathy, and they create a global telepathic network. And that is the future of humanity. Moreover, they can teach normal people to be telepathic, and they do that. So now their teachers and caregivers are telepathic, and they talk to each other, and they even teach lessons in silence using telepathy. I did my research on telepathy since 2009, so I have sufficient experience to believe that it is true from my personal research. Carl Jung is a pioneer in psychic research and also in telepathy. And in 1933 letter, he said that the existence of telepathy in time and space is still denied only by positive ignoramuses. And especially revealing our experiments with telepathy in dreams, dream telepathy, and psychic clear vision in information transfer in dreams. The key researchers here are Diane H. Powell, which is the main academic researcher who stands behind the telepathy tapes, and her book ESP Enigma, the scientific case for psychic phenomena, is one of the key books about telepathy and psychic phenomena, and she explains how brain anatomy contributes in psychic phenomena. Dean Radin wrote Real Magic, and so many other good books. And he is one of the main researchers, and he summarized big paranormal research, parapsychology research, especially in telepathy and information transfer. And Rupert Sheldrake, morphic resonance, and other books are on that topic. So these are three main names in academic alternative science about psychic and telepathy research. So from that, we basically understand that there is a pretty good body of evidence that actually the materialistic worldview is incomplete, and the only solution to solve the consciousness problem is through paranormal research and actual quantum physics. Here I just highlight the ideas that DNA has contributes to body in two ways. It goes through producing RNA, and it goes through producing proteins, and then proteins make up the body. So from physical matter to the body, DNA creates the body, and changes in DNA sequence create changes in the body. And also there is a second idea proposed by many researchers, including our collaborator Richard Miller, that DNA produces biofield and interfaces with biofield, and biofield also programs the blueprint of the body, the shape of the body. And then we start looking more closely at DNA, we realize it is not naked, it's not pure double helix, it is wrapped around nucleosomes, and that also, so this is like a pretty well-studied model of a nucleosome. This is a pretty well-studied model of tetranucleosome, and these are very orderly structures. The DNA is very orderly. Nucleosome is a little bit less orderly, but pretty orderly, pretty regular. And tetranucleosome is the largest orderly structure of DNA. It's still pretty small. It's in the order of under 20 nanometers. It's pretty small. But after that, DNA is wrapped into chromosomes in a similarly disorderly nature, in similarly disorderly way. So there is no known larger orderly structures of DNA. And that's basically, after that, it becomes a soup, which is disorderly. But so we're trying to find the logic in that chaos of DNA. And it seems to be the key to understanding that life is based on combination and dance between order and chaos. So also in understanding this picture, it's important to realize that DNA is hydrated, and it's very essential to understand that water structure is tightly linked with the structure of DNA, and it's pretty much impossible to decipher the role of DNA resonance in the body and biofield in the body without understanding the structure of the water. And we'll have speakers today that speak about that. So water structure, like we all know snowflakes, but the mystery here, how that part of the snowflake is synchronized with that part of the snowflake. And that, I think it's still a mystery. I don't think anybody asked that question and addressed that question. Somehow they are coordinated, and there is no known mechanism that would explain that coordination, because local conditions here and local conditions here are different. So, but there is a coordination and synchronization between 6 branches of the snowflake. And that's another mystery to be addressed. Next, we started looking at water structure around DNA. And I stumbled upon the idea of Gerald Pollack, that a flat water structure, which he calls easy water, the DNA imprints on these flat structures, imprints its own structure, helical structure. So I started looking and discovered that this flat structure was first described by Lippincott in 1969, and it's based on discovery of deragging of polywater about 10 years before that. So there are several names, including Dziriagin. Actually, there was someone before Dziriagin. I'm sorry, I don't have in front of me, but in the chapter, I spell out this history. It's possible to find the history how polywater was discovered, and then it was sort of discarded by mainstream science, and it went into being one of the prohibited, discarded parts of the science, as usual. So what is important about this polywater is it's in Lippincott that it is not exactly perfect. It has gaps and canyons, and these canyons allow it to be electrically neutral. If you fill the gaps with structure, it will become negatively charged. So there is a dance which is continuous of building and dissolving that structure. So that dance is super important. That is a slide from my 2023 talk where I say that DNA from one cell talks to another cell through waves, and these are nucleosomes, and basically the structure of DNA is important to transmit the wave information, and then it can go from structure to waves, and then from waves back to proteins to structure. Next slide is that nucleoplasm is very thick. There is liquid water, there is chunks of crystal water, liquid crystal water, DNA, and it's 1% DNA, about 20% of protein, and the rest is water. So it's very thick and it's constantly moving. And this movement is actually shown on the video. It's well known. It's called chromatin reorganization. It's a sped up video, but it's still very fast. And it is a whole nucleus. That part you already seen before. And I see that someone already is sleeping. Hey. All right. So here I looked at I tried to decipher the idea of Pollock that layers of polywater are shifting. And I realized that these shifts can imprint, the sequence of DNA can be imprinted on the layers. So I created that model where layers of polywater are actually perpendicular to DNA, and they're shifting as in pin tumbler lock. So as pin tumbler lock has a key classical key has peaks and basins, the pins in the pin tumbler lock go up and down. And the same idea is that as DNA changes its shape dependent on double helix and the sequence, the layers shift right and left. And in the first layer, it is a stepwise movement in polywater. It can move in six positions, and the next step it can move to another total. I think if you count these dots, it's after two steps it can move into 18 or 19 positions, I think. And that's just another illustration of shifts in polywater, how they reflect the DNA sequence, not only double helix, but actual sequence. That's a nucleosome, and showing that parallel DNAs probably are bound by structured layers of polywater. And the idea that polywater is built around negative structures is also belongs to Pollock. He studied that well. So that if you put like charged surface, then polywater is built around this surface. Here I expand the idea of disagreement between the structure of DNA and structure of water. The polywater is hexagonal, has hexagonal symmetry. DNA has hexagons, and it has a bit of hexagonal symmetry right here. No, right here. Hexagonal symmetry in DNA. So these are hexagons. But if you look at double helix, right here, its symmetry actually is 10.5 terms, 10.5 steps per turn of the double helix, and in two turns it's 21 steps, 7 multiplied by 3. So in two turns, one turn and another turn, it's 21 steps. So there's 21 steps per two turns, and 6 6 steps per turn, they disagree between each other. And that, I believe, makes DNA uncomfortable in water. It's, on one hand, it's hydrophilic, and it is bound to water, and it exchanges protons with water. On the other hand, I think it vibrates back and forth, spinning back and forth, trying to fit in water structure. And that, I think, is the basis of life. It's the main basis of energy, and that's why we are warm-blooded, to get our to produce our 36.6 degrees, to increase the vibration of DNA in water. And because DNA is charged, that creates oscillating electromagnetic fields in water. So the whole dance in the nucleus is a dance which is acoustic, electric, and electromagnetic with with lots of frequencies. So that's, I think, is one of the keys to DNA resonance. That is just another illustration of how DNA symmetry is disagreeing and conflicting with water symmetry. Next, I jump in forward. I'm close to the end. Yeah, jumping forward. That's a larger DNA structure, and I propose that repetitive sequences. There are multiple families of repetitive sequences called transposons. They are not positioned in DNA in order. It's a semen disorganization. They are pretty random in DNA. I suggested that positions of these repetitive elements called transposons in the DNA sequence create a bigger structures of DNA by coupling them together. I called it homological coupling. So it doesn't really matter. The idea is that it doesn't really matter what repetitive sequence it is, as long as you have two similar sequences in the DNA strand, they will attract to each other. And there is some quantum theories suggesting that similar structures, similar sequences of DNA would attract in water and couple. So it's a weak coupling. It's a new force. It's not well described, but the idea is of couplings creating the new structures. And because of Brownian motion in water, it's a live dance of forming and dissolving structures, larger structures of DNA. And next I went to test this using genetics data, known genomics, published genomics data. And the method is called chromatin conformation assay. It's published. There is a lot of those, many of those, not a lot, but I would say hundreds of data sets published, good quality. What they do is they freeze the structure by adding formaldehyde and linking all the proteins together. So this structure is frozen in time. They take cell culture, add formaldehyde, freeze the current structure of DNA. Then they cut it with a restriction enzyme, or it's called micrococcal nuclease enzyme to cut. Then they ligate using ligase, and that creates combined ligation junctions and sequences that combine together. So they were far away from each other, but due to ligation, they are now captured together, and that is sequenced using standard inexpensive sequencing. Once you do that, you know what stood in a cell, in live cell, what was near each other. And that's allows you to test whether, test that hypothesis, that repetitive sequences come together. So hooray, I found that they do come together. I learned with, first it was done with a hired programmer, but since artificial intelligence became good in programming and inexpensive, I learned how to program myself using AI. So it's pure vibe programming. I speak in English and it does the programming. I run it on my own server in Amazon instance, in Amazon virtual machine on Linux. And for the last 4 months, I was doing that. Finally, the results are being submitted, hopefully in today or tomorrow. What you see is that different transposons called MIR-B, L2B, 3N, L2C, 3N, they are enriched near the ligation point where things were ligated. And moreover, they are specific. So similar transposons come together. So basically that idea is verified. I cannot prove it's causal. It could be just a coincidence that in pieces that come together, these transposons are specifically coupling together, but now I have a metric which actually demonstrates that. And that's homotypic specificity. Homotypic means the same family, say MIR2B, is coming together in two strands. So if there are two pieces of DNA, if they are linked, if they're contacting each other, the chances that they occur together are elevated near that junction, near the contact point, and they fade away to background level far away. So I think that's I'm pretty confident that's real. And it's reproducible in biological replicates, and it's reproducible between different individuals. So the coming together or going away from similar. So these transposons that like to come together, these are transposons that push against each other and actually repel each other when they're together. They don't like to be in contacts. And this idea of not liking being together, I think, is also very, very essential. So in many cases, the signal comes from the sequences repelling each other specifically. So the L2C 3N transposon repels and prevents the DNA from contacting. So I think these are two factors going together. This is a slide going through all my presentations. There is a lot of frequencies starting from infrasound, which is fractions of hertz, to very high frequency, which is X-rays and ultraviolet. And these all frequencies which are important. I ran out of time, but I should highlight the idea that quantum entanglement in biology is super important. It's pioneered by Philip Ball and explained that much of health and biological signaling goes through quantum entanglement, and that quantum entanglement is spread through radiation and Brownian motion. And the reason sun is healthy and is therapeutic for us is because the rays of sun are coherent, and that coherence is absorbed by us by eyes and skin and makes us coherent and healthy. So coherence is given to us through quantum entanglement, through light. Same thing with healthy food. Coherence is the key, as many other speakers would probably will mention that coherence and quantum entanglement are key to health and also to consciousness. The last couple slides I will show that we are forming global telepathic network. It's already happening. It will happen through natural ways, through normal people learning telepathy, and through artificial ways, through using telepathic helmets. It's called synthetic telepathy, and that's also coming. Currently, it's done through Neuralink, which is surgical, but there are already helmets which are optical, and they also work through quantum entanglement. So it's possible to actually connect to technology and become telepathic electronically. Check out Richard Alan Miller's paper from around 1980, 1990 about synthetic telepathy. So that happens, and another key change for joining us in one telepathic network is artificial intelligence, which also will help uniting us into a more coherent system. It's a bumpy road, but I think it is inevitable. And I will end with the idea that it's super important to understand ecology of DNA vibrations, of DNA resonance, that the whole biosphere is one united vibrational system, and that this ecology is strongly affected by electromagnetic means and also by genetic manipulation of plants, animals, and humans. So the genetic manipulation of plants and animals is fully in progress, and there is a lot of genetically modified organisms. And genetic manipulation of humans is illegal in many countries, but there are countries where it's still not outlawed, and it's so easy to do that it's very likely it's already happening. And it's mentioned by some of the military leaders that genetic creation of soldiers is also attractive to them. So genetic manipulation of humans right now happens without understanding the vibrational ecology, and that will damage the collective vibration of humanity. So discovering of the DNA resonance and vibrational ecology is essential for survival of the species. And again, I highlight the site dnaresonance.org. You can find the whole chapter there. And again, this is a Frere model suggesting that we are a mirror of the universe, fractal mirror of the universe. And I thank my collaborators, Ivan Savelyev, Oksana Poleskaya, Michael Rempel, Ekaterina Erin Preza, Nelly Zarianova, and Richard Alan Miller. And these are my past workers. My collaborators, Irina Konstantinova and Vadim Gushin, who already are crossed over. And this is my contact. Thank you very much. I think I used all my time. I guess I'll take one question if you have.
May I have a question? Yeah.
All right.
Yeah. Thank you very much, Max, for your very lucid and very interesting presentation. I would like to emphasize that you have a fractal rendering or fractal mirroring from internal mind to external world. Uh, Although it is also known yet there is a mirror brain that or mirror neuron that work such in such way, but I think uh, we could look to other field. For example, Stephen Lehar. If you already know, Stephen Lehar work about uh, his Gestalt, yeah, Gestalt theory of uh, space, that he hypothesized that uh, our brain actually create the space here, create the space by consciousness.
I agree with that.
Maybe it's, yeah, maybe, maybe it sounds more like a back layer. There is no reality, but the space itself is created by the brain. But I think it is interconnected. Yeah. Like you say that the consciousness affect the brain, then the brain affect the consciousness. And the second, I appreciate that you mentioned that we should try to uh, discourage people, especially researchers, to modify human DNA without concern of vibrational ecology and consciousness. Yeah, and what I'd like to ask is uh, one, uh, how the interaction between consciousness and brain that people may call it neuroplasticity, can be used to help, for example, in the case of dem- dementia or Alzheimer, yeah, because many people are growing old and uh, maybe there is a way to train again the brain to rejuvenate. Yeah. I had a relative that have a case of dementia and Alzheimer.
I got it.
Maybe,
I got it. Thank
yeah.
you.
And
It's a
the
big
second,
question.
oh yeah, okay, yeah.
I'm running out of time. I'm over time for 20 minutes, so I need to invite next speaker. But I wanted to highlight the idea of coherence, and that coherence is key to not only neuroplasticity, not only to shape of the neurons, but to inner health of neurons, and which is linked to health of chromosomes. And this is chromosomes in neurons, the chromatin in neurons and chromatin in the rest of the cells. It's damaged by environment, but also by the mind. It's both are important. So coherence in the environment is important of the food and vibrations, but also coherence in the mind is super important. So yoga, I just discovered that yoga is one of the great tools for bringing coherence, and the meditation in yoga and many other Tai Chi, Qigong, Reiki, and other cultures, other natural healing techniques are greatly help coherence in the mind and in the body, and I believe it will help the chromatin. But also technological introduction of coherence is also important. Finally, after the red light therapy was discovered in 1968, many years ago, but finally about 10 years ago there was green light from FDA in the United States. So now a lot of red light therapy devices are finally on the market. And I think it also works through quantum entanglement and bringing coherence to the body. So technologically, it's also possible to increase coherence in the body, not only chemically, but through waves. And I will stop here because my time is over. And I would invite the next speaker,